Today, we are diving deep into one of the most incredible processes in all of biology, cellular reproduction. This is it. It's the engine that drives growth, the master mechanic that handles all our repairs, and it's quite literally the reason any of us are here at all.
But let's start with a really interesting question. Just think about it for a second. Why are you, or an elephant, or a massive tree, made of trillions and trillions of tiny microscopic cells? Wouldn't it be so much simpler to just be one giant cell? Well, it turns out nature has some very, very good reasons for not doing that. And that's exactly where we're going to start. To figure out why we're not just one big blob, we have to understand the basic physics that cells are up against. It really all boils down to a simple problem of logistics.
Okay, so this image here explains everything. Look at those cubes. As a cell gets bigger, its inside, its volume, grows way, way faster than its outside, its surface area. You can think of the surface area as the cell's front door and all its windows. Now imagine your house suddenly becoming 1,000 times bigger inside, but you still have the exact same tiny front door. Yeah, you'd have a big problem, right?
That's precisely the jam a big cell would find itself in. Its front door just isn't big enough to get food and oxygen in fast enough to feed that massive interior. And forget about taking out the trash. All that toxic waste would build up inside. Plus, imagine the nucleus trying to send an urgent message to the other side of the cell. It would be like sending a letter by snail mail. So, here's the bottom line. To survive, cells have to stay small and efficient.
So if cells can't just keep getting bigger and bigger, how do we grow? How does an organism get larger? Well, nature came up with a truly elegant solution. Cells grow just a little bit, and then they divide. This beautiful repeating process of growing and dividing is known as the cell cycle.
The cell cycle is basically the life story of a single cell, a story that it repeats over and over again. It's how you grew from one single cell into the person you are today. It's how your body heals a paper cut. It is a perfectly timed sequence of events. Grow, copy the entire genetic blueprint, and then split into two. And as you can see from this diagram, the cycle really has two main acts. A cell spends almost its entire life in this big section here called interphase. This is all the prep work. You've got the G1 phase where the cell just grows and does its day job. Then you have the critical S phase, S for synthesis, where it duplicates all of its DNA. And finally G2, which is one last growth and prep phase. All of that leads up to the main event, which happens in this much smaller slice of the pie, mitosis, the actual division.
Now, during that long interphase, all the DNA inside the cell's nucleus is in a form we call chromatin. The best way to think about it is like a huge, messy ball of yarn. It's all unwound and accessible, which is great because the cell needs to be able to read the instructions on the DNA to live. But can you imagine trying to split that messy jumble of yarn perfectly into two identical halves? It would be an absolute nightmare.
So right before it divides, the cell does something amazing. It performs this incredible feat of biological engineering. You can see it right here. That loose, messy chromatin gets wrapped super tightly around these proteins, then coiled and coiled again until it forms these dense X-shaped structures that we all recognize as chromosomes. It's like taking that messy yarn and neatly winding it onto spools so you can hand out a perfect, complete set to each new cell.
Which brings us to the showstopper, mitosis. This is not some chaotic tearing apart of a cell. Oh, no. This is a highly choreographed dance, a cellular ballet, with every single step precisely timed to make sure that genetic material gets divided perfectly. And this ballet has four main acts: prophase, metaphase, anaphase, and telophase. So let's break them down act by act.
Act 1, prophase. This is where the stage is set. The chromatin we just talked about scrunches up and condenses into those thick, visible chromosomes. At the same time, this thing called the spindle apparatus starts to form. You can think of it as all the ropes and pulleys being set up backstage, getting ready to move the stars of the show.
Next up, we have metaphase. A good way to remember this one is M for middle, because that's exactly what happens. All those chromosomes are moved to the very center of the cell and line up in a perfect single-file line. This alignment isn't just for show, it is absolutely crucial. It's how the cell guarantees that the split is going to be perfectly even.
And now for the most dramatic part of the whole performance, Anaphase. The spindle fibers pull tight and snap. The sister chromatids, the identical halves of each chromosome, are yanked apart from each other. Think A for apart or away. They are now their own individual chromosomes, racing toward opposite ends of the cell.
The grand finale of our ballet is telophase. As soon as the two sets of chromosomes arrive at their destinations, they start to unwind and relax, turning back into that messy chromatin again. And then a brand new nuclear membrane forms around each group. So now mitosis itself is done. We have one cell, but inside there are two identical nuclei.
But wait, the job's not quite finished. There's one last step called cytokinesis. This is the division of everything else, the cytoplasm. The cell membrane pinches in the middle, tighter and tighter, until it splits the whole thing into two separate, brand new, and genetically identical daughter cells. And with that, the cycle is complete, ready to start all over again.
Now, as you can probably guess, a process this important and this complicated can't just be left to run on its own. The cell cycle has an incredible, sophisticated system of checks and balances to make sure everything happens just right and at the right time.
So who's in charge? Well, the main bosses are these proteins called cyclins and CDKs. Think of them as the managers on the factory floor. By themselves, they don't do a whole lot. But when a cyclin pairs up with the CDK, it flips a switch, activating it and giving the official go signal for the cell to proceed to the next phase of the cycle.
And this is how those managers do their job. The cell cycle has these built-in checkpoints, which are basically like a quality control inspection. At the first checkpoint, the cell asks itself, is any of my DNA damaged? Later, before division, it checks, did I copy all the DNA correctly? Then, during mitosis, it asks, are all the chromosomes attached to the spindles like they're supposed to be? If the answer to any of these questions is no, everything stops until the problem gets fixed.
So that leads to a really important question. What happens when this amazing, intricate control system breaks down? What happens if a cell just starts ignoring all those stop signs and divides uncontrollably?
Well, that is the very definition of cancer. It is, at its core, a disease of the cell cycle. It's what happens when those checkpoints fail, when the stop signals are completely ignored and a cell starts dividing over and over and over again. It's the cell cycle with the brakes completely cut.
This comparison really lays it all out. Normal cells, they listen to signals, they stop when they're told, and the whole process is neat and organized. Cancer cells are the complete opposite. They blow right past the checkpoints, they divide in a totally chaotic way, and instead of repairing the body, they just start crowding out all the healthy tissue around them. It's the same process, just running wild without any of the crucial controls.
So you see, the cell cycle is the very engine of our growth, our ability to heal, our existence. But when it goes wrong, the results can be devastating. And that leaves us with one last, huge question to think about. As we get better and better at understanding this unbelievable process, what happens when we truly learn how to control it? What could that mean for medicine, for aging, and for the future of life itself?